We'll do that. We start by taking our glass microfiber filter and we place that right onto the filter here. And this is magnetically sealed and that holds it in place. Here's our deionized water. We run 1 liter through. And then through vacuum suction, it will pull the water through this manifold into here through the filter. So that was half a liter. We run this twice. And the thought process is that if there's any contamination of DNA on the sides of our filter top here, it would be picked up in the DI water and would later be shown in the analysis. So we'd know that was dirty if it was picked up positive DNA. So all of these controls should come back negative. So our control paper will go into a separate container and we'll label that with today's date. And this is sample 34. Now that we've run the control, we can start with the real sample. So we put on our filter paper. Now the filters, when they come in, some of them can be very clean. Others will have a lot of particulates floating in here, and they start to settle on the bottom. So we want to make sure that that is evenly distributed, so we want to give it a good shake. Pour that in and start filtering. Depending on how dirty or clean each water sample is will depend on how many filter papers we use per sample. On average, we look around about 3 filter papers as our target goal for each sample. Sometimes it'll get up to 12 filter papers, I think is the highest that we've seen, and that can take quite a while. So we collect 120 samples, 2-liter samples each time we go out, and it can take upwards of 12 to 14 hours to filter the entire sample for the day. As you can see, this is a fairly dirty sample. The water is coming down, but it's very slow rate right now. So it's going to take a while to filter this complete sample. We'll use more filter papers for this, and that just takes up more time here. But all the samples should be filtered within 16 hours of collection. After that, the DNA starts to degrade and our sample becomes more difficult to see if there was DNA originally present. All the particulates have filtered out and are now settled onto our filter paper. So we'll just fold this up and keep it into this sterile container. Keep the lid on. We still have about 4/5 of the sample to filter still, so we go ahead and just replace the filter with a clean one. Replace the filter cap and start the process over. And we'll continue this until we've gotten through this entire bottle. Right now we're about halfway through. We'll turn the filter back on, and that's the process.